My interest in using very data-driven computational approaches to biology actually started with a chance interaction when I was a high school student with Cy Leventhal, who was a professor at the time at Columbia University. He actually started at MIT. I didn't know it at the time. He was a physicist who got very interested in biology. And I started working in his lab as a high school student. In fact, I dropped out of high school and went to work in his lab. I may be the only high school dropout on the MIT faculty, I don't know. And he really fundamentally believed that the hard sciences had a lot to contribute to biology. And at the time, that was a rare belief. And so when I went off to college, I went to Harvard. I studied chemistry and physics, even though I was very interested in biology, because I thought that that was a very firm grounding to understand biology from that perspective. And then as I was looking for research opportunities when I was a graduate student, I gravitated towards the most data-driven aspect of biology at the time, which was called structural biology. This is trying to understand the three-dimensional shape of proteins. Proteins or sequences a long string of amino acids, but they actually fold up into complicated shapes, and it's that three-dimensional shape that gives them their function. When they're stretched out into random shapes, they don't work at all. You can think of an egg white. The native state of the protein, the one that's functional, is what you get when you crack open the egg. But if you mix it with your blender, right, the proteins no longer function. They take on different shapes. And so that those stringy molecules no longer function. So I went into studying the structure of proteins using the technologies that are driven by physical approaches, X-ray crystallography, and also NMR. And spent a lot of time becoming interested in how the structures could inform us about the biological function. And that was around the time then that the whole revolution was occurring with the sequencing of the genome and new technologies. And I saw that this was a tremendous opportunity to take a data-driven approach, not just to how one molecule interacts with its target, but how all the molecules interact, and to really zoom out and try to understand the larger scale of biology in a data-driven way. And so as this field was emerging, this field has become known as systems biology. I was very fortunate to get a position at the Whitehead Institute as a Whitehead Fellow, which is a completely independent, carefree position, no teaching obligations, just research, where one can go pursue whatever one wants. And that's where I retrained myself in computer science and learned how to apply computer science to these large-scale biological data. I think one of the things that is important in trying to handle the complexity of biology is to take an engineering approach. Biology has been driven for many years by approaches from the more qualitative sciences, trying to get in your head a model of everything that's going on and try to make predictions based on your own rationalizations, which can be very powerful when one is looking at a small system. Typical figure at the end of a paper might have 5, 10 molecules involved, and when you're dealing with 5 or 10 molecules that you know deeply because you've worked on them, you can often make pretty good predictions without the use of any computer program, without any use of high throughput data about what's going to happen. But even in those cases, there are surprising results that have occurred. There are many examples where drug development has failed because that intuitive reasoning doesn't always produce the right answer. And so the key that we believe is taking this engineering-based approach. Engineers are used to dealing with complexity. Think about the number of parts in a jet engine or the number of parts in your cell phone. Engineering is about making predictable models based on the complexity of the system, but our quantitative knowledge of some aspects of it and our probabilistic knowledge of other aspects of it. Biology may be unusual in that in most cases we're dealing with probabilities, uncertainties about what molecules are going to do, but we can bound those uncertainties. We can have the computer make for us educated guesses, if you will, about what's likely to happen, and then give us probabilistic answers. It's more likely than not that this will happen or that. And another critical aspect of these engineering-based approaches is the feedback cycle. We make predictions, but we don't stop there. We use the results from those predictions, whether they're right or wrong, to improve our models. And then we have continuously improving models of the biology of a system that allows us to engineer better and better solutions to whatever problem it is that we're studying.